I have a few announcements before I introduce today's speaker. The herbarium talk on Friday, April 15th at 12:30 is Robert Griffin Nolan from Santa Clara University. And so that's at 12:30 this Friday. Essig brunch, there's a seat right here if somebody's looking for a seat. Essig brunch Friday at 10 is Ida Naughton from UC San Diego, and the title is a multi-species comparison of genetic diversity between island and mainland ant populations. I don't have the IB seminar announcement, but there is IB seminar tomorrow at the usual time at 12:30. And next week in here, Emily Richards. Where's Emily? Is she here? There, right there. You're giving your finishing talk entitled, "I Get By With a Little Help from My Friends. Adaptive Genetic Exchange Contributes to Rapid Diversification and the Evolution of Novelty in Pupfish." So that should be fun. Are there any announcements before I introduce Mallory?
So the last herp group is next Monday, right? Yes.
Is it next Monday or the Monday after? We just had her.
Oh, right. Sorry. Okay, forget it. Sorry, forget it. It's 2 weeks. I just know it's special because it's in Chambers and it's going to be at Becca's house. Yes. So, but that's you're right.
I want to remind everyone we have the MVZ retreat coming up on Wednesday of RRR week at Tilden. And thank you to all the students and postdocs and everyone who have signed up to speak.
Monday of our week, right?
Yes.
Yes. Okay.
Monday of RRR. Thank you. There are 3 retreats that I'm going to that week. One on Monday, one on Wednesday, one on Friday. Okay, anything else? Well, it's my great pleasure to introduce Dr. Mallory Ballinger for her finishing talk. So unlike in most situations, this is a fait accompli. She already has her PhD. And when I asked her about giving a finishing talk, she said she wanted to wait and do it in person, which I really appreciated because it's more fun than by Zoom when we can gather in person. So Mallory finished her PhD in December. And so today is really just a celebration, and you can join us afterwards in this room. We're going to have a little some sweets and some champagne. For me, it's bittersweet, and I mean this with all sincerity. I'm very proud of everything that Mallory has accomplished. She's been just fantastic from start to finish, so I'm very happy for her, but I'm also sad because I've so much enjoyed interacting with her over the years, and I'm gonna miss having those daily interactions. So normally when I introduce someone at their finishing talk, I try to dig up some dirt. It's a time-honored tradition, and tease them a little bit publicly. And I looked for some dirt on Mallory, and I couldn't find any. and this speaks to her truly professional demeanor. Always, she just I don't think for a second that there's no dirt. It's just that she's hit it very successfully from me for 6 years. So instead of teasing her and digging up compromising photos, I'm just going to stand up here and gush from the heart about how great Mallory is, and I'm going to embarrass you that way. And I want to begin. So I went back and I looked at the first email that I found from Mallory when she was applying to graduate school, and I was thinking, well, maybe there's something embarrassing in there, but no, it's a perfectly crafted email describing all of her previous work. It was a little bit fawning. She was telling me how fascinating my research was, which, the kind of thing you see in these emails. And really, the only thing I could find in there is, let's see. Is this on? Oh, just. All right. So here's that email. I don't expect you to be able to read it, but you know, it's perfect. It's grammatically correct. It's perfect. She describes her previous research experience, her interests. This is September 11th, 2014. The only thing that was a little funny was she told me she wanted to study under my tutelage. And I thought, you know, tutelage? All right, so you know, she's trying to use a fancy word. Whatever. And then she also told me that she got her undergraduate degree from Black Hills State University. And I thought, well, where the hell is that? I've never heard of that. And oh, well, this is me. I wrote back, and the only thing of interest here is that I said, let's set up a meeting by Skype. That seems so dated now. Nobody uses it, you know, just 6 years later. So, you know, I thought, well, where the hell is Black Hills State University? So it's in this town called Spearfish, which is, for those of you who don't know, is in the westernmost part of South Dakota. And for those of you who don't know, South Dakota is a state kind of right near the top, population of 14, I think. And so I thought, okay, well, so Spearfish, that's an interesting name. Let's do a quick Google search and see what's in Spearfish. And this is what I found. So you know, I thought, geez, that looks pretty cold. So then she went on in her letter and she said, from there she got her master's degree at the University of Minnesota. And I thought, okay, well, I've heard of Minnesota, and I know the University of Minnesota and you know, in Minneapolis, but no, Mallory went to the campus in Duluth. She didn't want to go to tropical Minneapolis. So there's Duluth, you know, way up there. And when you do a Google image search of Duluth, you find, and so I thought, okay, well, I'm seeing sort of a pattern here. Here's Duluth in the summer. And so, okay, so then Mallory came, and I suggested to her early on that she put together some ideas about what she wanted to do her thesis on. And Mallory, being Mallory, was very thorough. So, you know, sometimes when you ask students for this, you get, you know, it's like pulling teeth a little bit. You get a few ideas, maybe a chapter or two. And so Mallory gave me this, and I don't, again, expect you to read the whole thing, but there are 5 PhD theses here with several specific aims, all laid out. And I thought, well, you know, that's ambitious. And as I read through this, I noticed there was a common theme here. Temperatures, cold, Eskimo, cold. And I thought, okay, so that's what Mallory is going to study. And it's what she did study. And she's done almost everything on this, actually. There are a few things I don't think you got around to, but it's kind of fun to go back and look. So Mallory was one of those students who was incredibly independent. It's kind of a truism for professors to say that they learn as much from their students as they teach their students. And in Mallory's case, it was particularly true. She came here with a very strong interest in animal physiology and phenotypic plasticity. And I hadn't thought much about phenotypic plasticity, and it was really through her that I came to know a new literature that's become very important in our lab. So I want to thank you, Mallory, for being such a good mentor, for patiently explaining things to me when I didn't understand them, and really for teaching me a lot. She's published 9 papers, and including a part of her thesis, which was published recently in AmNat. It's a beautiful paper. I printed out the reviews, and I was going to gush more, but let's just say they were fantastic. And the editor went on and on about how important a contribution this was. She, in her independence, sought out others. So while she was here, she published a paper with Caroline Williams. While Mohammed Noor was here on sabbatical, she published a paper with Mohammed. So being independent and motivated has really served her very well. And, you know, I think above all, she's very, very curious as a biologist. And that's a great attribute that will carry her far. She's already a postdoc working with Zach Gompert. And so today she's going to tell us about some of the work she did while she was here. She has a number of tangents that she's not going to be able to talk about today, but that are going to result in full-fledged papers of their own. The last thing I want to say, I didn't want to forget to say this, is that among all the other things Mallory did, she did a long field season where she conducted a transect and sampled both Peromyscus and mus from Texas to, of course, Minnesota. And she asked her father to join her as a field assistant on that work, and he did. And that just touched me. So as the father of 2 daughters, I can only hope that someday they might ask me to do something like that. And so Mallory, it's been nothing but a pleasure to have you in the lab, and I won't take any more of your time so you can actually give a seminar, but take it away.
That was super kind. And now I'm not look like a professor trying to figure out Zoom. You think Zoom can see my situation? You can tell I've been on Zoom for the past year without having to like make anything meaningful. Okay. Well, again, thank you, Michael. That was very touching. And I made sure you couldn't find any dirt on me. So. I think it all works out. No, but I really enjoyed my last, I was going to say a handful of years here, but I think it's definitely a solid 6.5 years at the MUZ. And I made some memorable memories with you guys. And like Michael said, I probably did way too much for my PhD, but I just want to show you a few of the things that I did while here in the museum. And so I think if I think of a question that motivated my entire PhD, I know I had a lot of tangents, but the main question was, how do organisms adapt to new environments? And I think many of us in this room have this question in mind. And you can think of it in 2 contexts. In one context, the organism would stay put, but the environment around it changes. And this is a major goal of the Cornell Resurvey project here in the museum. And then the other context, which is what I thought for my PhD, was when an organism moves into entirely new different environments, when they colonize a new environment. And as you can imagine, going into a totally new environment exerts pretty strong selection, just because every access of a new environment is different, where something like environmental change, when organisms stay put, only a few parts of the environment changes. And so, as Michael mentioned, I usually think of plasticity first. And so when an organism first gets to this new environment, what's actually happening? And what I mean by phenotypic plasticity is just the ability of an organism to change its phenotype in response to the environment with no genetic change. And so there's a great literature on how plasticity may facilitate the colonization of new environments. And I'm gonna steal a fun little cartoon and illustration from one of my scientific crushes, Ava Fisher. But you can imagine an organism in an old environment, just chilling out, having fun in their old environment. And they're depicted as this gold medium-sized circle. But the new environment that they're going to go into, the adaptive trait is to become a big blue circle. And so plasticity can act in a few different scenarios. In one scenario, plasticity can get you all the way to become a new big blue circle. And so this would be an example of adaptive plasticity. And because that plastic response gets you all the way to the new optimum, it's buffering that change from selection. Additionally, you could also have adaptive plasticity where you kind of get pretty close to the new big blue circle, but you're a nice medium-sized light blue circle. And then selection will act to get you close to the new optimum. And then finally, you could just do something totally different, become a small red circle, which is an example of non-adaptive plasticity, but then selection will be pretty strong to get you to where you need to be in that new environment. And so there's an awesome literature describing when you would expect to have adaptive plasticity or non-adaptive plasticity. And I'm not going to get into that today, but what I want to focus on are these adaptive traits in a new environment that we think of, so these big blue circles. And in reality, much of adaptation, especially adaptation to new environments, involves complex traits. And so what are complex traits? We had to put an equation on it. Complex traits are just some phenotypes that are underlined by many genes and are highly influenced by the environment. And so as an organismal biologist, when I think of complex traits, I always kind of think of the organismal level, such as changes in morphology or physiology or behavior. And one of my favorite examples of adaptation to environments and complex traits are the high altitude peromyscus mice. These mice that live at high altitudes and hypoxic environments. There's a whole suite of changes that happen from physiology, even down to the molecular level, to allow them to adapt to these hypoxic environments. But complex traits are also at the molecular level. So gene expression is actually a complex trait that's highly influenced by the environment. And there's been a bunch of recent studies that ask about the plastic response of gene expression and how that aligns with the evolved response to a new environment. So I just highlighted a recent fun example by Shane Campbell-Staton looking at forest anoles moving into an urban environment and how plasticity aligns with that. But overall, I tried to, for my PhD, encompass both organismal and gene expression levels of complex traits and thinking about plasticity. And of course, when you guys think about complex traits and adaptation, you think of your favorite charismatic organism, which of course is the house mouse. House mice are very cute. I recommend going to INAT and, well, maybe not, there's some gruesome photos. There's some cute photos of house mice on INAT, which is where I got all these photos. And I'm going to argue that house mice are an excellent system to ask about how the environment influences complex traits and how those complex traits are selected for in new environments. And so one of the reasons why house mice are great is because they've recently expanded into a wide array of new environments. So thanks to European colonists, I guess, house mice have hopped on board and have colonized a whole bunch of new environments all around the world. And today I'm mostly going to focus on house mice that have colonized North and South America. But before I dig in, I'll just give you a roadmap of where I'm going to go. So the main question I'm asking is, how does the environment modulate the evolution of complex adaptive traits using house mice? And so the first vignette, I'm going to try to just tell you two simple stories about complex traits, but we'll see how that goes. But the first vignette is asking about how the environment shapes morphology, so phenotypic differences that we see in house mice. And then the second half of the story, we'll be looking at how the environment influences gene regulation, so expression plasticity in general. And then the great thing about gene regulation and gene expression is it's an intermediate phenotype. And so it kind of gets you pretty close to the genes that might be underlying some of these traits we're interested in. And in particular, we can try to pinpoint to some genes to try to connect the genotype to phenotype map. That's a major goal of evolutionary biology. And so I'm just going to hopefully tell you an aside about linking some of our candidate genes to signatures of selection in wild house mice. But first, let's start with some organismal biology. So as I mentioned, house mice have recently colonized North and South America in the last 500 years. The last few, they've only been here for a few hundred generations. And as you can imagine, they've colonized something from hot arid environments all the way to a tropical environment and everything in between. But as Michael mentioned, before I came here, I just finished my master's in Duluth, Minnesota, and actually experienced the polar vortex in Minnesota. And so I think about temperature a lot, and temperature is a major climatic selection pressure across North and South America, as you can see in this map. And so this just got me thinking, well, phenotypically, can we see if there's any variation in house mice that are collected from anywhere from Alaska all the way to the equator, if they show any phenotypic lines, which would hint at adaptation to these various environments. And so some of the more famous ecogeographic patterns are just clinal variation in body size and extremity length. So body size coincides with Bergman's rule, which is just the observation that body size increases as you move away from the equator. So mice from Brazil are much smaller than mice that you can collect up in northern Alaska. So these guys follow Bergman's rule, suggesting that house mice have adapted to cold climates. And I just want to say, I downloaded a bunch of house mouse data from the awesome database of BirdNet and did some filtering. So thank you, BirdNet, for a great data set. House mice also conform to Allen's rule. So Allen's rule is just the observation that as you move away from the equator, extremity length decreases. So mice from near the equator have much longer tails and ears than mice from up near Alaska. So again, conforming to Allen's rule, suggesting that they've adapted to colder climates. And so with these three phenotypic lines associated with Bergman's rule and Allen's rule, points to the fact that house mice have adapted pretty rapidly to these novel environments. But just from data collected from wild house mice, it's hard to disentangle what's actually genetic and which is environmentally influenced. And so that's what I was interested in. So what we did was, through a bunch of help from people, we collected mice from the ends of this latitudinal transect, so near the equator at 3 degrees south latitude, and upstate New York, which is 45 degrees north. And brought them back here to Berkeley. They've been chilling in the warm lab ever since. And we can ask, do we see differences in body size and extremity length when you bring them back to the lab, indicating if there's a genetic basis? And when we do that, we do see body size differences still persist in the lab after many generations, indicating a genetic basis. So New York mice are much larger than Brazil mice, 50% larger. Cute photos, I know. And then Brazil mice actually have much longer tails and longer ears than New York mice. Again, this is after many generations of hanging out in the lab. So we think that there's a genetic basis to this. Also, while here at the museum, I had the privilege of collaborating with Eric Riddell, who is a former postdoc in the museum. And I asked him to measure some thermal conductance between our New York and Brazil mice. And interestingly, New York mice lose less heat through their fur and skin compared to Brazil mice, again, highlighting that they might be adapted to a cold environment. And I'm not going to show you any of the physiology data that I collected with a friend and colleague, Lisa Tridell, but we measured liver mitochondrial respiration rates, and they do match the same patterns we're seeing where New York mice seem to be adapted to a cold environment. So I keep telling you this, mice, cold, they seem adapted, but how does the environment actually influence some of these traits that we're interested in? And specifically, how does a mouse that's been hanging out in a warm environment, you put them in a cold room, what happens? And so that's what we did. We found a cold room here in VLSB, a walk-in refrigerator, and we reared both New York mice and Brazil mice at room temperature and in the cold, and asked, how do these ecogeographic patterns of body size and extremity length change? And so what I first want to show you are body size trends. So I should have mentioned this earlier, but New York mice are always depicted as blue and Brazil as gold. And these solid lines are mice that were reared at room temperature, and the dotted lines are mice that were reared in the refrigerator. And so, as you can see, pretty strikingly, we obviously see differences in body size, like I've been telling you. But no matter what, if the mouse is reared in the cold, there's very little plasticity in body size. And so whether you're a New York mouse or a Brazil mouse, body size shows very little plasticity to temperature. And you might be used to seeing these kinds of results as a reaction norm. And then next we wanted to ask about extremity length. And so I'm just going to show you results for tail length, but we see the same results for ear length. So again, same patterns of solid lines are warm mice and dotted lines are cold mice. So if you just compare the solid lines, we again see that Brazil mice have longer tails than New York mice. But Brazil mice that were reared in a cold environment, their tails were highly plastic, and they grew much shorter in the cold compared to the room temperature. And in fact, they almost get to the length of a New York mouse tail, but not quite. And so we see a lot of plasticity in both New York mice and Brazil mice, but Brazil mice shows greater plasticity in tail length, where New York shows less plasticity, so they seem to be canalized. And again, we see the same pattern for ear length. And so just to summarize some of these fun organismal phenotypes of how the environment influences morphology, we see that body size and extremity length show different patterns of plasticity. Body size is canalized in both New York and Brazil, but New York mice are canalized for both body size and extremity length. But the plastic response of Brazil mice and extremity length does depict this blue dot, this blue circle that we talked about earlier, where you do see adaptive plasticity. And because they don't get all the way to the New York evolved response, we think that selection would act further to get them to the new optimum. So that was organismal phenotypes. And now let's switch gears and talk about gene expression. As I said, much of adaptation involves complex traits, and gene expression is a great complex trait that is very influenced by the environment. I think one of Michael's famous quotes is, if you just cross your legs, you get differential expression. And so how can we, said that many times. And so how can we decipher patterns of gene expression are due to genetics or the environment? And so I want to walk you through just this ternary plot that can band gene expression patterns into expression patterns that are controlled by genetics or the environment or a combination of both. And so I drew these cartoons, and like I said, we'll just walk through, but gene expression patterns that show a complete genetic response are, you see differences between two populations, but you don't see differences in expression across environments. And so regardless of environment, you see differences between populations. If a gene is highly influenced by the environment, you don't see differences between populations, but you do see differences across environments. And then of course you can have a combination of both, a classic genotype by environment depiction. And so in this case, you do see differences in expression between populations, and you also see differences across environments. And then depending on which population you're looking at, each population responds differently to that environment. So it's the stereotypical crossing of reaction norms. And because gene expression is highly influenced by the environment, you can see a bunch of patterns of gene expression that fall in the intermediates of all these three different patterns. And in particular, we're interested in genes that show expression that are highly influenced by genetics and the environment, because these are genes that we can try to get at the genetic basis of plasticity for. And so how can gene expression be controlled at the genetic level? There are two main mechanisms that we can think of. Gene expression differences can occur via cis or via trans, which is a lot of jargon, I know, so let's just walk through it. A gene that's regulated in cis means that there's a mutation, just like denoted here with this X, that is very closely linked to the gene that's being regulated. So this mutation could be in a promoter region or enhancer region, and that mutation directly affects the gene that it's regulated. In contrast, trans-acting mutations are encoded anywhere in the genome. And they, for example, what I've drawn here, it could be an amino acid change in a transcription factor. And then this transcription factor goes and regulates some other gene somewhere else in the genome. And so the difference between cis and trans is cis, you're close to that gene, and trans, good luck trying to figure out what's happening. So throughout the literature, we think much of adaptation actually occurs through cis regulatory changes, just because they're less pleiotropic. But both cis and trans do play important roles in adaptation in general. And so how does one actually determine if their gene expression patterns are controlled in either cis or trans? Well, it's really, really easy. You just make F1 hybrids. And so if you can make an F1 hybrid, I highly recommend to study gene regulation. And so why does it matter if you can make F1 hybrids? So if a gene shows differential expression between the parents, so for this cartoon, a gene is more highly expressed in Brazil compared to New York, and you want to know how that gene is regulated, just make an F1 hybrid. And the F1 hybrid, if it recapitulates the difference you see in parents, then you know that that gene is regulated in cis. So if you see differences in parents and that difference is recapitulated in F1 hybrid, that gene is regulated in cis. So just easy peasy. So that's what we did. We made a bunch of F1 hybrids by crossing a line from New York and a line from Brazil. And as I mentioned earlier, we also want to know how the environment impacts gene regulation patterns in general. And so we did the experiments that I've been talking about. We put mice in the cold and kept mice at room temperature. And then we sequenced a bunch of RNA from two tissues, brown adipose tissue and the liver. And these two tissues play really important roles in metabolism and thermal regulation. And I just want to highlight that they might actually be playing super important roles in our mice. So for the liver, I'm kind of using food consumption as a proxy for liver activity, but regardless if you're a New York mouse or Brazil mouse, all mice are eating a lot more food in the cold. And so you can imagine a lot of metabolic processes are increasing in relation to the warm environments. And then brown adipose tissue is my favorite tissue of all. Someone who thinks about the cold, but it's a heat generating tissue. So it takes white fats, it consumes it, and all those free fatty acids decouple the electron transport chain from making ATP to producing a bunch of heat. And so it's a really cool heat generating tissue that was first found in hibernators. But here, as you can see, New York in general has more brown adipose tissue than Brazil, but regardless, both Brazil and New York mice increased how much brown adipose tissue they're making in the cold, which is beneficial if you want to produce some heat. Okay, so we generated a bunch of RNA from these two tissues across parents and F1 hybrids. And so what does the data look like regarding gene regulation? So I'm going to walk you through some patterns of cis, trans, and hand wavy cis and trans. And so in all these plots, on the x-axis will be differential expression between the parents. So between Brazil and New York. And on the y-axis is differential expression between the alleles in an F1 hybrid. So if you see differential expression between the parents, and that's recapitulated in the F1 hybrids, what do we get? Cis, yeah. And so these will fall along this diagonal here, and I've denoted them in an orange color. But if you see differential expression in the parents, and that's not recapitulated in the F1 hybrid, then those changes, the difference in gene expression is due to trans, and so that those genes will fall along on this horizontal line in green. And then because gene regulation is complicated, you can get a combination of both cis and trans, and I'm not going to bore you with those details, but those will fall out along this axis in blue. And so if we just ask how gene regulation divergences between New York and Brazil, this is what we see, a lot of dots. But in general, we see a lot more of these orange dots. So regulatory divergence between New York and Brazil is largely due to changes in cis, about 10%. And this aligns well with previous studies in house mice. And like I said, genes that are regulated in cis are exciting because it's pretty close. We know that the genes being regulated, the mutations that are regulating those genes are pretty close to the genes of interest. And so I'm going to cycle back to that here in a few moments, but much less variation or much less regulation is due to trans, about 5%. And so that's great. We didn't know that about New York and Brazil mice, so that's exciting. But what I was interested in was how does temperature affect these patterns? And so if you look at a scatter plot from the warm versus a scatter plot for the cold, you can obviously see major differences. Just kidding. What we do notice is that genes that are regulated in cis in one environment are highly robust to the temperature change, and so they're cis in the cold environments. But what's changing the most are trans effects. So trans effects are highly temperature dependent and sensitive to the temperature. And so if you squint in these scatter plots, you can see that the green dots are changing the most, which intuitively makes sense. If you're a mouse and you get put into a refrigerator, a bunch of metabolic processes are going to happen. And so a bunch of transcription factors are going to be regulated and expressed and regulating other genes throughout the genome. So this wasn't too surprising. But what we really want to know are genes that show plasticity, but are also regulated in cis. And so I picked a gene to explain this, but if in the warm environment, This gene shows differential expression in the parents and the F1 hybrids. It's regulated in cis, and we want to know if that gene is changing and not regulated in cis in the cold environments, because these are the genes that could underline the genetic basis of plasticity. And so when we ask this question, we only find 14 genes that show this pattern, which again is not surprising given that cis changes tend to be robust to temperature. But still, 14 genes is pretty exciting. What are those genes? I'm not going to show you all of them, but I'm going to show you two of my favorite brown adipose tissue genes that play important roles in thermoregulation and adiposity, SCD1 and CDH13. And so what I want you guys to get from these reaction norms is that both genes are differentially expressed in the warm, and that's underlined by a cis effect. But that cis effect pretty much goes away or is diminished in the cold. And that's mainly due to the plastic response of Brazil. So New York shows no plasticity in response to the cold, so they're canalized, but the plastic response of Brazil is probably diminishing that cis effect. And this plastic response of Brazil is adaptive. It goes in the same direction as the evolved response of a New York mouse. And so seeing canalization of New York expression is cool. And we actually see that overall, if you look at overall differential expression in the parents, New York mice between warm and cold show fewer differentially expressed genes than Brazil mice. And so again, suggesting that they're more adapted to a cold environment. As if you remember, we also see this with morphology. So tails and ears of New York mice are canalized in response to the cold environment than Brazil. And so just to summarize briefly the environmental effects on gene regulation, what we found was that most genes, a lot of genes show plasticity, and that this plasticity is regulated in trans, and that New York mice overall tend to show patterns of canalization. But that if you put a Brazil mouse in the cold room, their plastic response tends to be adaptive, which is exciting. Okay, I want to circle back to the 10% of cis regulated genes. So like I mentioned before, gene regulation is an intermediate phenotype that gets you pretty close to the genes that might be underlying adaptation. And so let's try to connect this phenotype to genotype map and look at genes that are regulated in cis. Are they actually showing signatures of selection in wild house mice? And so if you take all of those genes that are regulated in cis and ask how they're enriched or what kind of pathways are enriched, they make sense. It's mostly it has to do with increasing your body size or increasing your lipid levels, which is great because of the tissues we sequenced and also because we see large body size differences between New York and Brazil house mice. And so you might be wondering, well, what are some of these genes that are enriched in these pathways? We see a bunch of cool ones, which I know you can read, but I just want to highlight four of my favorite. I have a lot of favorite genes, I know. But I want to highlight the leptin receptor, LEPR, and FADS2. So these play important roles in thermoregulation and adiposity, and they've been shown to be under selection in humans adapted to cold environments. But the other two, PCAT2 and ADAM17, are also really cool because in a previous study by Katya Mack, found these genes to be outliers and cis-regulated in mice that are found along the East Coast of North America. In the direction of the allelic expression pretty much align with the patterns we saw along the East Coast. So this is exciting that we're seeing some of the same genes in using a different method and also using some different mice. Okay, so you could take genes that are regulated in cis, figure out which pathways they're enriched for, and then you can overlap that with scans for selection. And hopefully you'll find some candidate genes that might be underlying environmental adaptation. And so one of the great things about being in the Nachman lab is there's a wealth of genomic resources with wild house mice collected across North and South America. And so we capitalized on exome sequencing of New York mice that are found in the wild, along with Brazil mice. And what I'm going to show you is selection that's been happening along the New York branch. And so Using PBS statistics, if that means anything to you, that's great. We can look for large allele frequency changes along the New York branch, which is indicative of selection. And so we find a bunch of outlier SNPs when we do that. And now we wanted to overlap those with genes we found that are regulated in cis between New York and Brazil. And when we do that, we find 64 genes, which are highlighted here in orange. Here are some of those genes. Again, if they mean something to you or if you're excited about them, that's great. I'm also excited about them, about this amylase gene. But I think you notice our friend BCAT2 is there again. And if you zoom in on BCAT2, most of the outlier SNPs are hanging out in the exon region, which just means that there might be some biology happening in this Manhattan plot, which is great. And so this is just a snapshot of trying to figure out some genes that are influenced by the environment, and that might be under selection in wild house mice. And I saved you from all the complicated things, but I can answer any questions you might have going forward. But overall, I think for my PhD, like I said, what really motivated my questions was how do organisms adapt to new environments? And I really wanted to plug the environment in this question. I think the environment can really influence the trajectory, obviously, of evolution and how some of these traits are selected for. And hopefully I convinced you to also think about the environment a little bit more. So now the fun part, I guess, of finishing talks is acknowledgements. I'm going to try not to be too emotional. I guess if I become emotional, I have to stop at the acknowledgements. But there are a lot of people to really thank. The science is fun to talk about and to show you guys, but honestly, none of that would happen without most of you in this room. And I can't acknowledge all of you because I only take pictures of my dog. And so if you're not in a photo with my dog, I just can't acknowledge you. But first I want to acknowledge a bunch of the funding that I've had the privilege of having for my entire PhD. But first I need to thank many people in the MBZ, especially these three people. So first and foremost, Terry. I think every time I came down to the prep lab with some mouse skeletons, I think she kind of was like, all right, here we go again. But thank you, Terry, for helping me skeletonize all those house mice skeletons. I know they made your day. I also want to thank Chris Conroy for his patience. I know I need accession, still a few more mice, but I'm going to get that done. But thank you, Chris, for making sure that happens. And then finally, I think my biggest thanks goes to Lydia Smith. Not only did Sylvia and I have more than 200 RNA-seq extractions and libraries to do, but also some extra because, you know, things happen. But none of that would have ever been possible without Lydia's mentorship and guidance and just like knowing everything about everything. And so Lydia, honestly, if it wasn't for you, none of this would have been able to happen. And I think I could speak on all grad students and that we're very thankful for you. So thank you, Lydia, for helping us. I also want to thank my committee, Craig Miller, Noah Whiteman, and Caroline Williams for pushing my thinking on not only just the results of my PhD, but in evolutionary biology in general. And I want to give a special thanks to Caroline. As an organismal biologist and a physiologist, when I first got to Berkeley, I fit right in with Caroline's lab, and she welcomed me and became a secondary mentor. And so thank you, Caroline, for letting me hang out with your lab and being mentored by you throughout my PhD. Then the Nachman lab, there's so many people to thank. But first and foremost, I need to thank Katya Mack. From day one, she taught me all things gene expression and gene regulation and is still teaching me things and is still holding my hand. But Katya, thank you so much for being a great friend, a great collaborator, and being patient with me as I wrap my head around gene regulation. It's been awesome. I also want to thank the people who collected all these house mice and brought them back to Berkeley. The postdocs Mike Sheehan and Felipe Martins. Obviously, without them, I wouldn't have have been able to put mice in a fridge, so thank you. I also want to thank Taichi Suzuki, who was the first one to introduce me to house mice in the lab and taught me all of his secrets on how to wrangle mice. And he is probably the best mouse wrangler out there. And so thanks to Taichi. And then finally, a special thanks to Sylvia. As I mentioned before, Sylvia, I wish you would have joined the lab much earlier so we could have talked about tails and cold room and mice in general, but it's been the 2 years I had with you was pretty spectacular, so thanks. And then I guess if you acknowledge the Nachman Lab, you have to acknowledge Michael Nachman. So right when I got here to Berkeley, we took a field excursion as a lab and some other people in the MVZ to the Eastern Sierras. And that's where Michael showed me all things mammals. And I had the privilege to teach mammalogy twice with Michael, and I learned so much from him. And I think, you know, we learned a lot from each other working pretty close during mammalogy. But thank you, Michael, for your patience and teaching me a lot about PopGen. I know I didn't really talk about PopGen in this talk, but thank you for being patient with me and letting me put mice in the fridge. That was fun. And then there are so many friends to thank, but again, as I said, I don't have good photo taking skills. And so I apologize if you're not up here, but there are so many people in this room that I enjoyed hanging out with, talking science and also just life, and you guys are awesome. But there are 3 people, four-ish people I wanna particularly thank. First is Lisa Trudeau, who I think is here on Zoom. Day 1 of grad school, Lisa and I met and hit it off and have so many scientific interests and life interests. And Lisa, you've been a great friend, and I can't wait to keep doing science with you no matter where we end up. Second, I want to thank Ben Karen, who I know you can't probably recognize me in any of these photos. I look different, but Ben, I met Ben in my 2nd year, and I think, you know, I was going through a rough time, but Ben was there to pick me up, and we've been pretty close ever since. And you've been an awesome friend, and you know, I wish I could be in Berkeley so I could keep hanging out with you all the time. Thank you, Ben. And then finally, if you're wearing a pin, I think you could probably thank Michelle and Emily for that. Or whatever else is happening out there that I don't know. Similar to Sylvia, I wish you guys got here so much earlier. We only had a few months pre-COVID to hang out, but I'm so glad that we hit it off and became friends, and we still hang out even though it's on Zoom now. You guys are the best. Molly and I love you, and I can't wait to keep being friends with you guys in the future. And then I guess finally, there's someone else I should thank, and I mean this wholeheartedly. I think the best thing to happen to me here at Berkeley was meeting Molly Womack, who's an amazing partner throughout life and also just a fun person to talk science with. And we get to be dog moms to these pretty cute dogs. So thank you, Molly, for being there, especially as I'm an anxiety-driven person. Thank you for your patience. And so I think I have to stop there, else like liquid will come out of my eyes. We can talk, we can start drinking.
So I'm sure Mallory will be happy to answer any questions people have.
So I'm just curious about the whether it's known what the source populations are of these mice in Brazil and New York, whether there's enough phylogeographic information, for example, to tell you this, and then whether or not they show the same canalization patterns in the source populations that you see in the descendant populations, or if this is something that happened in one versus the other.
Yeah, that's the best question, the biggest question, and I think Kennedy will tell you the history of house mouse colonization. We, at this point, we don't really know exactly where all of the house mouse source populations are from. So we can tell that New York is probably pretty closely related to Germany, but that's not true for Manaus. Brazil mice. So it's speculation at this point, but we're pretty certain that house mice from New York were sourced by a different population than what was sourced for Manaus. But that's, I think, the ultimate experiment to do would be to take a Germany mouse and put it in a fridge and see if it shows similar patterns to New York. But we just don't know where South American mice are coming from. And Kennedy can answer that question sometime in the near future, hopefully. But yeah, that's a great question. I don't know. Yeah.
These morphology experiments where you're like putting the mice in the fridge, how much do you kind of think about if there's a difference between adaptive plasticity of like, oh, I want my tail to be smaller, so I'm gonna like make it smaller, versus just like I'm stressed out because it's cold, so I'm not growing as much?
Yeah, no,
How much
that's,
does that matter, I guess?
yeah. So for tail length, we know that temperature does directly affect the development of actual tail vertebrae. And so there is an aspect of just like biology and physiology with the response of tail length. Like you could put any mouse in a fridge and their tails are always going to grow shorter. And one of the reasons I didn't want to do the opposite experiment where you put mice in a really warm incubator is because once you start increasing the temperature, more things start to go awry due to stress. And so it's hard to disentangle like meaningful biology from just like stress in general. But I mean, in general, if you see a population like New York that doesn't show any plasticity, then you can compare that to the plastic response of a naive population like Brazil and kind of make the correlation that the plastic response is adaptive in that regard. But yeah, there's definitely some stress happening in the Brazil mice in general. Yeah, but
Awesome talk. So it looks like with the tail length and both New York and mouse were growing in the cold, you actually had more variance in the tail length than New York mice.
that's because they collapse the sexes together. So there is sexual dimorphism and body size between males and females of both populations. But for that reaction norm, I just kind of threw the sexes together. And so that's why you see a lot of variation.
So my follow-up question now is, have you explored differences in variance between the two populations? There's in either the phenotypic data or the gene expression data, there's just a bunch of predictions about how naive populations might have more variance when exposed to a novel environment.
Yeah.
Or adaptive.
Right. Just like phenotype, like more variation at phenotype level. Yeah. So in some regard, New York shows a little bit more variation, where Brazil is pretty tight, which could also correspond to the expectation that if you're from, you know, a variable environment, you would show a little bit more plasticity. But I think I would have to collect more data for the population level to kind of get at your question. So right now, most of this was line specific and not necessarily population level data, but that's, so I could go to like the VertNet data and see at a given latitude, do you see more variation at higher latitudes than we do. But I think that fits the alternative hypothesis that more variable environments might select for more plasticity.
Yeah, or well, we can talk about.
Yeah, mostly.
So as you talked about the thermal conductivity data, and I was curious if you measured it in the mice that you put in the cold environment afterwards to see if that was something that was being selected on.
Yeah.
or-
So we initially did like a pilot of skins of mice that came from the cold environment, the fridge, and there's very little plasticity in thermal conductance, and so I think it's associated with body size. It's just kind of like canalized in regards to a cold response. But we do know from previous studies that like fur insulation does increase in response to cold, but just not in our house mice. Yeah. Cool. Yeah.
Super cool, awesome talk. And I guess in the context of genetic assimilation and flexible stems, I guess you're seeing the opposite of what that predicts. That in Brazil, in terms of the ancestrally, these mouse were probably cold adapted. I don't know if you want to talk more about that.
Well, I thought you were to say something about genetic assimilation in the New York mice.
Oh, is that what you're saying? Yeah.
Well, I mean, I'm like tiptoeing around even using the word genetic assimilation because ultimately I want to take an ancestral mouse and put it in the cold and ask about plasticity. But if we think that the initial plastic response of New York mice when they landed in New York showed adaptive plasticity, and then over time selection has selected on that phenotype, and now the response is canalized, then that would be assimilated. But you're thinking of it in terms of Brazil mice.
Yeah, 'cause I guess it seems like ancestrally the Brazil mice are probably from a cold environment. So you're seeing a
They're from like a
gain of-
Mediterranean, we think like Mediterranean, Western Europe environments. And so there's definitely been selection in both directions, even though I mostly catch this in the cold. Yeah.
I know this wasn't specific to your study, but I was thinking about like humidity versus aridity. Like that's got to be, I mean, it's definitely going to be drier in New York than it is in Brazil, I assume,
Yeah.
for most of the time. And so how does that seem to affect the sizes and how will they do?
Yeah, so I'm a little biased towards temperature as being, more of an intense selective pressure, but Noel asked about aridity between Tucson mice and Edmundson mice in regards to kidney function, and we do think those two populations have adapted to differences in humidity and aridity. But the reason I'm leaning towards temperature for New York and Brazil is just because they follow those ecogeographic patterns that were suited in differences in cold temperature or warm temperature. But yeah, you're right. Even though Brazil doesn't show much fluctuation in temperature, they do have a rainy season, and maybe, yeah, there's something with humidity that could be driving some of these patterns. I think a lot of these climatic patterns are also correlated with each other, but yeah, I don't know what to expect if it was all driven by humidity, what kind of ecogeographic patterns you would expect to see. But yeah, that's a good question because more than temperature is varying between these populations. So yeah, Valeria.
Thank you for the thought. I was wondering if you saw, or I don't know if you have explored it or not, but a convergence with mice that live in the mountains, for example, in Peru or like in the Andes. Like if you see also in the cold temperatures the same things affected. I don't know if you have explored it or not.
Yeah, comparing mice found at high altitude and in the cold. Is that what you're?
Yeah, because like it's also cold. It's also high altitude,
Yeah.
but it's also cold. So in difference of the Amazonia mice as well.
Yeah, so Libby just published a paper looking at wild mice along elevational transects in Bolivia and Ecuador, which, as you mentioned, the high altitude populations are not only, having to adapt to hypoxic environments, but also cold environments. And it'd be great if someone in the Knockman lab could catch some high altitude house mice and bring them back here, and we could do that experiment. But I would think that there's probably some overlap in genes that you would find, just because all of these traits that we're interested in are complex, and so many genes will probably overlap between the two. But yeah, you can find house mice at high elevation. Yeah, I know there's some questions on Zoom, but maybe one more question, then I'll
Yeah,
look to Zoom.
I was wondering more about that. So I know that the temperature really affects individual tail vertebra development. How does that affect your cartilage development?
Yeah, so it's not as interesting as the tails. So if you shine a light behind a Brazil ear, it's way more vascularized than a New York ear. And I think a New York ear, this is all qualitative. It seems to be more thick as well than a Brazil ear. And so I think regarding Allen's rule, there's just a lot more heat being lost in Brazil ears just due to the high vascularization and thinness of the cartilage. But it's hard to get, yeah, I just want everyone just to like do gene expression of tails and ears in response to environment, because I think there's something interesting for sure going on. But yeah, in some regard it would be different because there's no vertebrae in yours. Yeah. Okay, I'm just gonna see if there's any questions on here. No, they're just saying it was great.
Just Ava Fisher raised hand.
Oh, Ava.
All right,
Yeah.
here I am to ask a question.
Hey, Ava.
So I have a philosophical question, which kind of goes along with the genetic accommodation. Do you think that in the next 40 years of your successful career, you could re-evolve the plasticity in the New York mice?
Can you repeat the question?
She's wondering if one would be able to re-evolve the plasticity of New York mice. I would want to just start from a European mouse population to do that. But are you asking like if I took a Brazil mouse or a North American mouse?
No, I'm asking, even though the New York mice are not responding to being in a warm climate, do you think over time you could get that response back? Like if it is in fact assimilation, could you make them into Brazil mice basically?
I think here's a philosophical answer. I think it's easier to go from small to big than big to small. So I don't think so. I think they're constrained by being big and adapted to a cold environment. But that's just mine. I have many thoughts on this about it, but yeah, I think it's
Call me later.
Other questions? Well, if not, please feel free to join us immediately afterwards for some goodies in here. And let's thank Mallory once again for a great talk.